Hey guys, Mr. P. In this video we're going to talk about the last stage of aerobic cell respiration, which is the chemiosmotic phosphorylation or the oxidative phosphorylation pathway. And so to begin our discussion of this last stage, we have to understand what has come before, and there are videos about all of these stages. But aerobic cell respiration, which is the form of cell division in which O2 is present, aerobic means oxygen is available, we are going to navigate this entire pathway. Again, all of this occurs in and around the mitochondria, but glycolysis kicks off this process. Remember, glycolysis is an anaerobic process, meaning it has zero O2 demand. Glucose is turned into 2-pyruvates. In the process of converting glucose to pyruvate, we produce a little bit of ATP, but we also produce these NADH molecules. In fact, we produce 2 of them through the glycolytic pathway. We then navigate the prep step, or transition into the prep step, which is also called the link reaction, in which pyruvate is turned into acetyl-CoA. In the process, we are also producing an additional 2 NADH molecules. Entering the Krebs cycle, we're going to take the acetyl-CoA and we're going to transition it through the intermediates like malic acid, oxaloacetic acid, succinic acid, and various other intermediates. And in the process are going to produce some NADH and FADH2. We are also going to produce a byproduct, which is CO2, which we release to the atmosphere and produce a little bit of ATP. But the purpose of the Krebs, as you recall from the Krebs video, is the production of 6 total NADH and 2 total FADH2s. Now I'm highlighting these blue molecules, the molecules in blue, because these are the molecules that are entering this last stage to supply ATP synthase with the needed electrons and hydrogen ions in order to pump out large amounts of ATP, which is the overall purpose of cell respiration as a whole. Now, there are a few ATP molecules that are made throughout the process, but the bulk of the ATP is made during this process. So we need to talk about how that ATP is made and what the role of these NADH and FADH2 molecules are. So the first thing we need to do when talking about ATP synthase and the role of this last stage of cell respiration, we have to understand how NADH works. NADH is the high energy electron carrier. It is converted from NAD+. So you'll notice by looking at this diagram that there is a relationship between NAD+ and NADH. This is the NAD+ cycle. NAD+, let's think of as an empty taxi, which is able to accept passengers. NADH is the full taxi after it has received its passengers, and its role is, just like any full taxi, to take the passengers to where they need to go. NADH is going to be used to shuttle the passengers, which would be the electrons and hydrogen ions, from either glycolysis, prep step, or the Krebs, to where they need to go, which is always the ETC, or electron transport chain. Now, if we focus on what this looks like, let's think of, again, NAD+ as this empty taxi. Now, all empty taxis are not fully empty because there is always a driver. So this driver's seat, if we were looking at the top view of this taxi car, is taken by the driver and therefore is not conducive to a particular passenger. When that taxi, which is NAD+ in this case, navigates to the passenger acceptance phase, right? It goes to where the passengers are. All of a sudden the passengers show up. The passengers in the NAD cycle is a hydrogen ion and 2 electrons, and so those passengers are going to bond to NAD+, converting it into NADH. Now, there is a positive H+ ion and 2 negative electrons that are going to join NAD+. The reason NAD+ is positively charged to begin with is because it will bond an additional positive and 2 negatives. If we start positive, bond a positive, now we are positive +2, and then we add our negative electrons, pulling it down -2 to a neutral charge, which is why the NADH drops the charge to neutral. NAD+ is converted to NADH, obviously because the H is the hydrogen that is bonded to this taxi, so it becomes NADH. Now that molecule is going to convert itself into NADH. It has accepted its passengers. It turns off its vacancy light. Notice it changed to red in this diagram. It no longer can accept passengers. And now it is going to head to the ETC, or electron transport chain, where it will drop off those passengers. Again, it will carry one hydrogen ion, 2 electrons, and notice it is plus H+, so it shuttles an additional H+ that isn't technically transported in the molecule. It kind of just follows, closely associated with it, dropping these passengers off at the electron transport chain. We will get into how those are specifically used in a minute. NAD+ is then going to go back to where it can accept more passengers and shuttle more hydrogen ions from glycolysis, prep step, and Krebs over to the ETC where they are eventually needed. So the NAD cycle converts NAD+ to NADH back and forth, back and forth. It shuttles the hydrogen ions and electrons, which are the passengers, to where they need to go. Now, do not forget about the redox reactions. Okay, reduction is gaining electrons. So NAD+ will be reduced by gaining electrons, turning it into NADH. NADH is oxidized, meaning losing electrons, because it gives up the electrons where they are needed, which is the ETC. Before we get into the actual intricacies of chemiosmotic phosphorylation, we need to talk about one particular component of it, and that is ATP synthase. ATP synthase is an enzyme that is embedded in the inner mitochondrial membrane. Its role is to transport these hydrogen ions through the channel protein down their natural concentration gradient from where they are high concentration to where they are low concentration. Again, we've talked about cellular transport mechanisms, we've talked about diffusion, we've talked about facilitated diffusion and passive transport. H+ ions are in a high concentration in this inner membrane space, which is the space between the outer membrane up here and the inner membrane here. These high concentration H+ ions are dropped off here by NADH, which is again that taxi car. They are going to flow through this channel protein from where they are high to where they are low. And as they flow through this channel protein, they're going to power the phosphorylation of ADP and phosphate and turn it into ATP, which is our high energy cellular compound. That is the compound that the cell uses for energy. Once these H+ ions flow through and the ATP has been made, they are on the low side and will, as you'll see later, be picked up by our final electron acceptor, which is oxygen, or pumped back to the high side to maintain that concentration gradient. But on this particular slide, we need to know how ATP synthase works. H+ ions moving through the channel protein, phosphorylating ADP, which is smashing a phosphate onto an ADP, turning it into ATP. This is the ultimate goal of cellular respiration and is definitely the goal of chemiosmotic phosphorylation. So if we talk about this stage of cellular respiration as a whole, you will notice that there is NADH. That is the compound we just talked about. NADH is our high energy electron carrier. Not only does NADH come from the Krebs cycle, which is also called the citric acid cycle, which again occurs in the matrix, which is inside of this inner mitochondrial membrane. NADH can also come from the prep step. It can also come from glycolysis, but regardless of where it came from, it is going to come in, it is going to drop off its passengers, which are a pair of electrons and a hydrogen ion. Those hydrogen ions will be shuttled to that inner membrane space. Where they accumulate in this space in a high H+ ion concentration. So we'll just go ahead and put a bunch of these. H+ ions are constantly being added to this space, and so they are accumulating in this high concentration gradient. NADH is constantly flooding this particular membrane with H+ ions and electrons because they are constantly coming out of the Krebs, glycolysis, and the prep step. Those NADH molecules are going to come to the electron transport chain, which is what this set of membrane proteins are. electron transport chain. The hydrogen ions, again, are being pumped to this inner membrane space. The electrons are going to travel from this protein to this protein to this protein to this protein to this protein, and as they reduce the next protein by being added to the next protein, they are giving off a little bit of energy at every step, and that energy is the energy that is used to pump these H+ ions from this low side back to the high side. If you recall from our membrane transport mechanisms, pumping of ions or movement of materials against a concentration gradient in general is called active transport, and it requires energy. This energy comes from the electrons that are being sent down this electron transport chain, which is maintaining this concentration as a whole. It is important that we maintain this concentration gradient because we will talk about the purpose of ATP synthase in a minute. But before we talk about ATP synthase, as these electrons move down this electron transport chain, they are eventually going to be picked up by our final electron acceptor. The final electron acceptor is oxygen. The role of oxygen is coming in as we inhale. O2 is going to pick up this pair of electrons, which is navigating the electron transport chain. When oxygen picks up the electrons, it's also going to pick up a pair of hydrogen ions, and those three components, the oxygen, hydrogen ions, and electrons, are going to come together to produce a water molecule. That water molecule will then be released to the atmosphere during aerobic respiration. Again, we breathe in O2, we breathe out CO2, and we breathe out water vapor. This water vapor comes from the electron transport chain. The CO2, in one instance, comes from the citric acid cycle. Both the CO2 and the H2O will be expelled through our exhalation every time we exhale during aerobic respiration. We inhale to breathe in fresh O2, and we exhale to release the byproducts of the cell respiration pathway, which are the water vapor and the CO2. Now, H+ ions are accumulating in this inner membrane space. The reason they are accumulating is because it is very small volume, and so it is easy to maintain a high concentration gradient. As these H+ ions are accumulating in this inner membrane space, they are then going to flow through this ATP synthase exactly like we explained it in the last slide. As they do that, they will power the phosphorylation of ADP and inorganic phosphate. Those two things are going to come together and are going to combine to produce ATP, which again is our high energy compound that is required for a lot of cellular activities. Once these H+ ions get down here to the low H+ ion concentration, again, they will either be pumped back to the high side to maintain that gradient, or these H+ ions, once they are moved, will join our final electron acceptor and again be released from this entire system as water. Looking at cell respiration as a whole, again, this is where this entire chemiosmotic phosphorylation pathway occurred. It requires the constant flow of NADH and FADH2, which is coming from not only glycolysis, but the prep step and the TCA cycle, or Krebs. All of these NADH and FADH2 molecules are coming into chemiosmosis, where they are dropping off their pair of electrons and hydrogen ions. These are the reactants that are going to help pump out not only the H2O, which is a byproduct of cellular respiration, but produce a ton of ATP. In fact, there is 32 to 34 total ATPs made during this process alone. If you learned something, give it a thumbs up, subscribe to the channel. We will see you later.